Sorry, I was busy conversing. That's what the main reason for these things are for. Otherwise, you can just listen to people talk. But I like the talking bit. The talking bit is really interesting, where you get to discuss ideas. My name is Jack Glibert. I'm a professor of surgery at the University of Chicago. I'm also director of the Microbiome Center there, but I have many other hats as well. And that's because I'm essentially a microbial ecologist. Right? I actually deal with bacterial communities inside any environment on Earth. And those environments can be really interesting, everything from deep down in the bottom of the ocean, all the way up to the top of Mount Everest, and then all the way in between to the lakes and rivers around Chicago, into agricultural sites. And that gives me a unique perspective, a perspective on how microorganisms work, how they interact with each other, and how they interact with the environments in which they find themselves. And remember, for all of you people who are terribly humanocentric, this planet is a microbial planet. They've been here for 4 billion years. Anything you can see with your eyes has only been here for about 500 million. So realistically, you are trespassing on already occupied territory. And I know Americans are good at dealing with that, so that's not a problem. But in this case, it's really interesting, right? How do we translate that basic component, the basic science behind how these microbes manifest and their ability to evolve and adapt to this environment? How do we translate that and implement it in healthcare? And so I'm very lucky. The guy following me is a visionary. I'm very lucky he's a visionary because he's one of the people who got me into the Department of Surgery at the University of Chicago. I am not an MD. They still do not allow me to cut people open, which I find rather archaic. However, I'm more than happy to be there because what I'm doing is bringing ecology into medicine. And ecology is a systems biology approach, right? It's using our understanding of how things talk to each other and how those conversations can manifest change in an environment. And that is really, really important. Because if you want to change something, you've got to figure out what you've got to tweak in order to change it. As we are hearing, and precision medicine is one of the key focal points for this idea, that the ability to change somebody has to start with an understanding of how all the pieces are connected. And you're a massive connected systems mess, and that is central to our research area. You're a meta-organism. You are an ecosystem of ecosystems. You're like a little planet. It's just like revolving around, and many people, especially the surgeons I work with, they'll understand that. I mean, their own little ego planets. No offense, John. But you have 40 trillion little people living inside you. Right? You can call them people if you want. I think they're some of my best friends are microbes. But there's 40 trillion of them living on your skin, on every mucosal surface, inside your lung, in your urogenital tract. Anywhere you can get to, there's microbes. Right? And they figured out ways to adapt to this environment. And this environment's very similar to most other animals, warm-blooded creatures on the planet. So we can look at other animals and see similarities between how they respond to their microbial world. Interestingly, they have around 7 million genes, independent genes, different genes that code for different functions that we don't have. So our little 27,000 human genes, with all of the regulatory elements that go along with it, pale in comparison to their genetic superiority, right? You can tell which side of the line I lie on. And that's really important because that gives us 7 million unique ways to manipulate the system that is you. And we think that that's very important because the system is heavily influenced by which types of microbes are there and what they are actually doing at any given time. Where does your microbiome come from? It comes from your mother, mostly. That first initial interaction is very important. This is a principal coordinate plot. All the dots are microbial communities, and the closer those dots are together, the more similar those microbial ecosystems are. So up at the top, you can see yellow dots. Those are samples taken from a swabbed around inside the mouth. We extract the DNA from those swabs. We look at which bacterial species are there, and then we compare how similar those ecosystems are. And mouths are generally quite similar to each other. Is there a plural for that? Yeah, I guess it is. These are like a number of different women, and you can all tell all their mouth microbiota are the same. Down in the bottom left in red, that's the vaginal microbiota, and over in blue on the right, that's the skin microbiota. So mouth, skin, and the vaginal tract are very different microbial ecosystems. And whether a baby is born vaginally or through a C-section determines what kind of microbes they first encounter. And so the bacteria that colonize and immediately stimulate the immune system of a neonate is dependent upon the route they take into the world, which is really interesting. Now, 3 months later, there's no differentiation. If a 3-month-old, you present me with that 3-month-old, I could not tell from its microbiome whether it was born cesarean section or vaginally. But that's less important because its immune system responses appear to be differentiated. And we know that children born via C-section have elevated risks of various immune-related disorders, such as asthma, allergies, eczema, but also, interestingly, things like obesity and depression, which don't appear to be immediately related to how you enter the world. But there are other trajectories there that could be relevant. So this leads to you being microbially unique. And every single one of us is microbially unique in our own way. I can tell which one of you is sitting on the chair you're sitting on based upon the bacterial community that you leave behind on that chair. More about that later. But this microbial uniqueness is very important because it enables us to stratify populations based on responder-non-responder rates and various other components in a way that we could never do before. It gives us an entirely new palette of tools in which to look at why our patients differ in response to a particular treatment strategy or a drug or what have you. That's very important. That gives us a new tool place. And even identical twins have a unique microbiome. So why one identical twin might get obese and the other one might not could actually be a function of the bacteria in its gut. And why the one might respond to a drug and the other might not might also be an influence of the types of metabolisms those microbes engender inside the body. And that's really important to understand. We think that we've been eroding the microbiome. Our modern-day lifestyle is not exactly conducive to having a diverse microflora. And this is because 150 years ago or so, we figured out that they cause disease, and so we've been on a war for 150 years trying to eradicate them. Rightfully so. If you went back 150 years, there were 50 million of us dying of tuberculosis every single year in Europe and the United States. That's a huge number of people. Death was so overprevalent that if your child died, you would take a photo of the dead child next to your living children and put it on the wall. I mean, that's pretty morbid and macabre now, but back then it was commonplace, right? We've forgotten that. We've forgotten how prevalent death was and the rule that microbes had over us because of that disease spread. However, we may have gone a bit too far. In our modern-day society, we're plagued by a number of different disease states which lead to many different ideologies that we can't quite figure out. You know, everything from elevated risks of cancer and inflammatory bowel disease all the way through to asthma and allergies, anxiety, autism. You can play the alliteration game here. Alcohol dependency, interestingly. Alcohol's next to breast milk on this picture. That's maybe not appropriate. Even diabetes. They are significantly elevated when we bring people into the environment, which is our nice, warm, westernized American world with access to plenty of sugary, high-fat foods and comfortable environments, right? What's going on there? We think that the microbiome and the immune system, the immune phenotype it engenders, could be extremely important in that. In fact, this is a list of some of the 399 ongoing clinical trials focused on different components of our health that are currently ongoing that have microbiome as a component, right? So all of these are under investigation. And that doesn't mean that microbiome causes all these disease. A change in your bacterial community, and remember you have almost 10,000 different types of bacteria in every single one of you. That's an immense diversity, right? And understanding that is incredibly complex, which is why we need ecologists, not surgeons. No offense, John. I'll say that a lot. But that's really important, right? There's a lot of them in every single one of you. And it doesn't mean that if they change, you're going to get one of these diseases. But alterations in what they do can affect other things inside your body. And that can lead to a cascade of events which might be prevalent or lead to the development of a particular disease, or exacerbate existing conditions. So understanding it is beneficial, even if it may not be the primary cause. So here are some examples. Bacteria on your skin can metabolize your sweat, releasing volatile compounds that can attract and activate the biting habit of mosquitoes. So we work with the army in order to try and figure out how to interrupt that relationship so that mosquitoes, when they find you, don't necessarily bite or reduce the biting efficiency. So bacteria are, the bacterial compounds that they release when they digest it are attracting and causing biting from these mosquitoes, so it's very important. Whether Tylenol produces, by its degradation in your gut, a toxic compound which can cause hepatic diseases, is relevant because microbes are the ones that are doing that metabolism. So whether you have a likelihood of developing liver cirrhosis associated with Tylenol activity is dependent upon which bacteria you have in your gut. There you are, and you've got an immediate thing, right? If we can figure out, or we know, if we can develop a test to identify in a patient whether they have that particular compound, if they want to take Tylenol as a long-term strategy for pain control, and they're positive for this, it might not be a good idea, because long-term manifestation might cause liver disease. This is a picture of two flies humping, which is also the name of our new band, which John and I will be talking about. Now, I can change the mating preference of a fly literally by swapping the microbes between one fly and another. So if I take the poop from one fly and I put it into the second fruit fly, the second fruit fly will now fancy the fly that the first fruit fly fancied, which is it's an interesting tongue twister, but it's also fascinating because I can change the behavior of an animal. And that is an animal, complex being, right? And that proves or suggests that I can have the same effect on humans, leading into an entire plethora of neurobiological disorders and relationships, which we're currently investigating along with many of our colleagues at University of Chicago. Everything from Alzheimer's and autism all the way through to depression, anxiety, and other neurodevelopmental issues. So we want to put the microbiome in your medical records, not because we want a job, but because we think it's vitally important to understand how this bacterial community inside you is changing, because if it changes, other things can change too. So most people will recognize the figure or know this study. This is a study based on a very ancient remedy from China called yellow soup or dragon soup, where, depending upon your dialects in your region, whereby you would take the stool from an infant pure of spirit and mind and mix it up into a broth and feed it to someone who had diarrhea. And the idea behind this was that you would alter the energy balance of the body from one that was disease-promoting to one that was health-promoting. We're not talking about energy balance or qi here. We're talking about changing the ecosystem from one that doesn't function properly to one that does function properly through a fecal microbiome transplant. So for a recurrent C. diff, we would maybe get a 30% success rate with constant vancomycin treatment and bowel lavage. With the infusion of donor feces, so feces from a healthy donor into the colonic cavity, we get almost a 94% success rate. Very small study, this. Only 16 people in each wing. Kind of crap, really, pun intended. But now there have been over, well, a little over 200,000 studies with fecal microbiome transplant for recurrent C. diff done across the globe, a lot of them in Australia, interestingly, and quite a vast amount here. And the success rate's still over 85%. So that's pretty good. That suggests that it does something, right? And we know what it does. The new microbiome, a damaged microbiome in the gut, doesn't degrade or doesn't produce the right metabolites from the metabolism of the bile acids pumped into the intestine. Some of those secondary bile acids produced by the right kind of bacteria actually inhibit C. diff from germinating, right? So it's in a spore form, and it can't release from the spore form if those chemicals are around. Now, in a damaged gut microbiome, the C. diff can grow completely happily because those secondary metabolites are not being generated. But if you put the right microbiomes back in, the right kind of bacteria in there, they re-energize that metabolism, and you get those inhibitory compounds back. So that's pretty interesting. That suggests we have a mechanistic understanding of that relationship. But there are many, many other factors that are in play. So what do I think has been happening in the world? I'm interested in exposure, and we used to be exposed to a lot of bacteria. Now we're not exposed to very many because we play inside and we live inside about 90% of our lives. And we kill everything as well, cleaning it ad nauseum, good and bad microorganisms. This has reduced the exposure our bodies have and our immune systems have to the microbial world, which we think has led to an acceleration of allergic diseases, both food allergies, asthma, and skin allergies. We think that by adding bacteria back into the exposure of a child, for example, we may actually be able to significantly alter their susceptibility to the activation of those autoimmune conditions. So we're very interested in that relationship. One of the studies we published last year in New England Journal of Medicine focused on the Amish and Hutterite populations in the US, working with Carol Ober and Ann Sperling at the University of Chicago and Donna Vercelli over at Arizona. We were able to disentangle the elements which might differentiate two populations of people who have two very different lifestyle exposures. Now, a little background. The Amish and Hutterites both come from Eastern Europe. Then they migrated through Europe, one through Sweden, the other through Switzerland. And then in the 19th century, to escape religious persecution, they came over to America. The Hutterites set up shop in the Dakotas, and the Amish set up shop through Pennsylvania, Ohio, and Indiana, right? Now, they live very similar lifestyles at home. They both don't like iPads and iPhones and electricity. I can bring my child to this farm in order to show them that you can actually physically survive without having an intravenous iPad relationship. And the kids on the Amish farm have a different relationship to the Hutterite farm. So while at home everything's quite normal, the kids in the Amish farm are living on the farm. Literally 20 feet from their front door is a barn containing the horses and the cows. And they're actually heavily involved in the working at a farm. So they have a very strong work ethic, and the kids, from pretty much the first time they can, are actively involved in the farm life. The Hutterites have a very different relationship. The men, and only the men, get up in the morning, get in a buggy, ride to an off-site farm where they change into their work clothes, do the work, get back, change into their house clothes, get back in the buggy, and go home. The kids, and ostensibly the ladies as well, the older women, are not allowed to actually work on the farm, so they never get that kind of exposure. So boys and girls of a certain age are never exposed to the farming environment, and the differentiation here is that the Amish have only 4% asthma in their population, and the Hutterites have between 25 and 35% asthma, right? Significantly above, almost 2 to 3 times the national average. They both have the same genetic predisposition towards the disease. So in this instance, a genetic test wouldn't predict which group would develop asthma. So therein lies the crux. But their immune system does. So if we look at their immune responses between the children in the Hutterite population and the Amish population, and the Hutterites here are in red and the Amish are in blue, we can see very big differentiation. For example, there are more neutrophils running around inside the Amish blood. There are lesser eosinophils running around inside the Amish blood. Interestingly, those neutrophils in the Amish are also much younger. So if you don't know what a neutrophil is, the neutrophil is like a little scout. It runs around, latches onto a foreign object and goes, hey, immune system, there's something nasty here. We need to get rid of it. And then the immune system sends along the little Pac-Man. You can tell I have young children. Comes along and it munches up, the Pac-Man being a macrophage, munches up the neutrophil and the foreign object. Done, dusted, everything's groovy. Right? And when you're exposed to a lot of foreign objects, your neutrophils are constantly being regenerated because they're constantly being munched up. So they never get old. And as we know about when things actually get old, they get a bit grumpy, right? I'm now over 40, which means that I'm super grumpy. Yeah? John's pointing out the fact that he's just turned 40 as well. And this is a problem. We've actually seen this not just in asthma. I mean, these kids have elevated levels in the Hutterite population, elevated levels of these aged neutrophils, which are very sticky and can overreact to the stimulus of a particular antigen. But we also see it in people with sickle cell disease. Young children developing vaso-occlusive crises in urban environments where they're not exposed to very many microbial antigens actually seem to develop a much older repertoire of their neutrophil cells, which leads them to having much greater stickiness, binding up the sickled erythrocytes and leading to a lack of oxygen penetration into the surrounding tissues, causing extremely painful events. Very similar relationship. An autoimmune disorder. We go in to look at people with sickle cell disease who are living in a more rural environment. They do not have that a significantly higher level of vaso-occlusive crises. So something's going on there. To test this hypothesis, we took dust from the rooms, the bedrooms. We snuck into their bedrooms at night like the Grinch. But we took dust from the bedrooms of Amish children and Hutterite children, and we literally exposed it intranasally into the epithelium of a mouse model that had been given a sensitization to evalbumin. So a little bit more evalbumin, or a big dose of evalbumin, and these mice will have an eosinophilic reaction. However, if we intranasally expose them to Amish dust, magical Amish dust, ooh, see, got that little boom there, magical Amish dust, then the mice do not have an eosinophilic reaction to the antigen, which is great. They're actually protected against the allergy, right? The Hutterite dust doesn't have the same effect. Interestingly, there's a dichotomy there. If we go into, some of the Hutterites have asthma and some of the Hutterites don't. And if we go to the homes of the, we take dust from the homes of the Hutterite children who do not have asthma, it's also protective. So there's something about that exposure which is protecting the children and also the mouse. And so we're now disentangling that. We found several antigens and we found several metabolites which appear to have a protective effect in vitro, and we're now testing that hypothesis in vivo in our animal models. And then potentially, in maybe 2 or 3 years, move forward with a clinical trial to determine whether we can reverse that clock or prevent the development of the disease in a population of vulnerable children. We also know that microbes can make you fat. One of my colleagues used to weigh 385 pounds. He lost 113 pounds by going on a traditional Chinese whole grain diet for a period of 9 months. He looked at his microbiome before and afterwards, and before there was one bug that was quite abundant, like 2%, and by the end it had nearly disappeared to complete absency. That bug was Enterobacter cloacae B29. When he grew up this bug and stuck it into a mouse, the mouse changed shape. The mouse developed a significant increase in white fat cells in adipocytes, and that led to even on the same calorific intake, with the same activity, which led to a differentiation in its, obviously, metabolic level. So one organism appeared to be able to trigger obesity in this animal, and we're not sure why, but that's a very interesting paradigm, right? We have a couple of hypotheses, and we've done a lot of work at UChicago to figure that out. We know that this organism, when given the right kind of food, will actually break down that food and release hydrogen sulfide very locally. That hydrogen sulfide can permeate into the bloodstream across the epithelial barrier and can actually interact with circadian clock genes in the various organs of the body. And so what you get then is you get a decoupling of the circadian rhythm of your organs from the circadian rhythm of your brain. Interestingly, the same thing happens when you go between time zones, right? So if you flew right now to Shanghai, you'd be 12 hours out of your time zone. Your organs would have a different sense of time to your brain, because your brain would be being triggered by the light-dark cycle outside, and you actually are more likely to put on more weight for the same calorific intake. So if you ate exactly the same food, your body would be processing it differently. So we think this microbiome, this bacteria, produces a similar response with a chemical adjuvant to that relationship. Cancer also shows a microbiome profile. This is a great example of lifestyle changes. In the green and blue, we have Japanese living in Japan. In pale blue, we have Japanese who have moved to Hawaii and have adopted a Western lifestyle. And then in red and white, we have Caucasian populations living in Hawaii. And this is the rate of prostate, colon, stomach, and breast cancer. And as you can see, prostate, colon, and breast cancer all significantly increase when they stop eating the traditional dietary lifestyle that they have in Japan and move over to a more traditional Western lifestyle where those diseases are more prevalent. Interestingly, stomach cancer goes down. These people are taking more prophylactic antibiotics, which are wiping out the Helicobacter pylori, which triggers the stomach cancer, right? So there's a relationship there, give and take, if you will. And it proves to us that we have an issue. We also know that germ-free animals tend to show a significant reduction if they have a mutation, say an APC min mutation. They have a significant reduction in the development of adenomas and polyps associated with colonic cancer, suggesting that microbes may play a role. And indeed, when we look at human populations, on the left here we have a family tree of bacteria. And the blue bacteria are all bacteria that are enriched in people without colon cancer. And the red organism is one that's only one enriched in Caucasian populations with colon cancer. It's called Fusobacterium nucleatum. And we can actually see that organism, you can't really see it here, but in the right picture we have the actual tumor tissue, and we can see when we stain it with red, the bacterium co-associated with the tissue. And it appears to be one of the only bacteria that directly co-associates with the developing tumor, and it modulates beta-catenin signaling, actually hiding the tumor from the immune system. We also know from work at University of Chicago that we've been able to actually alter the susceptibility, or sorry, the efficacy of a given immune blockade therapy treatment, alpha PD-L1, in an animal by augmenting it with a probiotic. So in this case, on the graph on the left, in blue we have a tumor growth associated with an untreated animal with a melanoma injection. And then in the open circles, we have the effective reduction in the tumor volume associated with alpha PD-L1 immune blockade therapy. And then in purple, in exactly the same level of activity as the alpha PD-L1 antibody, we have bifidobacteria. A bifidobacterial probiotic, something you can buy in Walgreens, right, seems to have in the animal a nearly significant association with tumor volume reduction. Now, whether that translates to humans isn't, as you all well know, hopefully, there is a very, very poor rate of success in translating stuff from mouse to humans, but it's a very interesting finding, and one that a company is actually, Evelo has licensed that technology from the University of Chicago and invested now over $60 million in trying to develop therapies associated with it. We know that when we combine the two of them, I don't know where my thing is, but that's the black line at the bottom. Combine the two of them, you see a significant increase in the reduction, or a significant reduction in tumor volume again. So it's great. The microbiome is all around us. This leads to complications. When you're trying to get rid of it, you can't. You can't sterilize it. Your clothes are a really crappy barrier to microbes. So everybody in this room who tries to pull down their sleeve and like open the door to the toilet, it doesn't make any difference. Trust me. This is like loose fishnet stockings to bacteria. They're really, really, really tiny. They pass straight through, right? So you can stop doing that right now and just accept the fact that you're constantly exchanging microbes with the world, and you're going to get used to it, right? And that opens up some interesting paradigms, because when we do epidemiological studies looking for disease-causing organisms in a particular environment, I don't know, like a hospital, it's very interesting to try and identify whether those microbes are causing disease. We did this for 365 consecutive days at University of Chicago's Center for Care and Discovery, and we found that there were constantly organisms which have the potential to cause disease in the exposome, the exposure environment of every patient. But most of the patients weren't getting sick. Why is that? What is it about what happens to a patient in a hospital and their exposure to microbes which could cause a difference? I light aside for 30 seconds. We can use these microbes for microbial forensics. Look at the kitchen counter up there on the left. That's the top right-hand image. The x-axis here is time. The y-axis is the percentage of bacteria found on that surface which originated in one of the 3 people and 3 dogs that lived in this home. Person 1 is a 25-year-old man. Person 2 is his dad in orange. And person 3 in yellow is his mum. So you can see this is a gender-imbalanced home because the vast, and sometimes 100% of the bacteria found on the kitchen counter originated with the mother, which is a great example of how I can use this for forensic approaches, right? In fact, we've done that extensively, and we now have a project with the Department of Justice led by Jared Hampton Marcel down here in the right-hand side with the pretty hairnet and mask on, looking at whether we can use microbial forensics over a 10-year program to improve the success rates for accurate or relevant identification of a perpetrator. Because right now, too many innocent people are being put in jail because of misidentification. This is a great example. I'm using this strategy to put people, to identify individuals in a complex environment. So we can definitely use it to try and stratify patient populations into responders and non-responders. So just a very last thing, I'm going to introduce, essentially, Al Verdi's work, because he's been my mentor and somebody who has opened my eyes to the possibilities that microbes could play a substantial role in health and disease in the surgical operating theater. So in looking at what happens to a microbe in an environment, I always tell the residents in Al Verdi's lab that they don't have one patient on the table, they have 40 trillion patients. So let's say we cut out a section of gut. We do 2 things. We allow oxygen into the gut. Some of the microbes don't like oxygen. It stresses them. Then we actually pump antibiotics into the bloodstream. That antibiotic pulse kills off a lot of bacteria, but it also stresses the ones that are resistant. Then we stitch up the gut, forming an anastomotic junction. This does one final thing. It allows the body to start repairing itself, sucking nutrients out of the gastrointestinal cavity and causing nutrient depletion, which causes stress because the microbes start to starve. They change phenotype, selecting for ones that can grow on the wound area, releasing collagenase and breaking down the collagen matrix, leading to leak. So these are microbes. Some of them are endogenous into the microbiome. Especially if you live a pretty, let's say, unhealthy lifestyle, you're more likely to have some of these organisms inside your gut during surgery, which will become so stressed that they try to protect themselves by gaining their environment back and altering it. And unfortunately, that has negative consequences for your health. And so I know Alvedi's going to speak more about that, so I won't talk about it anymore. So precision medicine can go three ways. We can either feed the microbes with a prebiotic to cause an ecological shift to remove a bad actor. We can add a probiotic, which causes an ecological shift to remove a bad actor, or we can use targeted antibiotics. The prebiotic and probiotic strategy is reasonably well established and is where the industry is heading at the moment. And the targeted antibiotics are more complicated, but definitely under development. So just leave it there. I have a book out. If anyone wants to read it, you can. And I'll stop. Thank you.